تكلمنا في المحاضرة السابقة عن موضوع الimmunoglobulins, their structure, what is the constant region and what is the variable region, and identify different classes of immunoglobulins like immunoglobulin G, M, A, D, and E. Now we will talk about another lecture which is about the immunoglobulin types, isotypes, allotypes, and idiotypes.
Determinants present on the antibody molecule. You might have heard about isotypes, allotypes, and idiotypes. So we'll see what are these antigenic determinants. Now, to understand this topic, it is important that we know the basic structure of antibody, which we have spoken about in one of the previous videos. So I'll just put the link on the screen in case you guys want to have a look. Determinants. Now, to understand this topic, it is important that we know the basic structure of antibody, which we have spoken about in one of the previous videos. So I'll just put the link on the screen in case you guys want to have a look. Now, we know that antibody is actually a glycoprotein, right? And we also know that when it comes to antigen, proteins are very good antigens, right? So proteins or the antigens have the ability to actually induce our immune system. And antibody is also a glycoprotein. So if you think about it, antibodies also should function as potent immunogens. That means it should be able to induce the immune system, right? Logically, if you think about it, since antibody is also protein, a glycoprotein, that should function as immunogen, which induces the immune system. All right. There are specific regions on the antibodies that can actually induce the immune system, and those specific regions are called antigenic determinants. So these antigenic determinants, or these specific sites on antibodies, they fall under three categories. That means on an antibody molecule, there could be such three types of antigenic determinants, such three kind of different regions that could induce the immune system. And those are isotype, allotype, and idiotype. So we'll see all three of them one by one. It's interesting and it's very easy to understand. So the first one is the isotype. Now, the meaning of the word iso in Greek is same. Okay. Now, we know that every antibody in humans, let's say for example, we have 5 different types of antibodies, right? We have IgG, we have IgM, IgA, IgD, and IgE. Right? We have 5 different types of antibody in our serum. All of us, all the humans, we have these 5 types of antibodies. That means all of us would have specific genes that codes for each specific immunoglobulin. Right? All of us should have these specific genes that codes for these immunoglobulins. And we have talked about this in the antibody structure video, how these antibodies, how do we label different antibodies? That is based on what type of a heavy chain we have, right? Let's say, for example, if we have heavy chain delta, that means that immunoglobulin would be IgD. Let's say, for example, I have this is the heavy chain. This one is the heavy chain. I have a gamma heavy chain. That means the antibody is IgG. If we have a heavy chain mu, that means the antibody is IgM. So we have total 5 different types of heavy chain for 5 different types of antibody. Versus that for light chain, it can be either kappa or lambda, right? Either kappa or lambda. Any one light chain can be there. Furthermore, in detail, if you see, there are again, types of this light chain and some of the heavy chain. Like, say, for example, lambda can also be lambda 1, 2, 3, and 4. Gamma heavy chain is gamma 1, 2, 3, and 4. And for alpha, it is alpha 1 and alpha 2. So we have all this light and heavy chain coming together to give us 5 different types of antibodies. And those different types of heavy chain and light chain combination decides what type of antibody it is, right? So that means there are specific genes that codes for that specific heavy chain or that specific light chain. Either it is kappa or lambda, or if it is heavy chain delta, heavy chain alpha. You have specific genes that codes for that particular type of heavy or light chain. Okay, and it is to be again more specific, we are talking about the constant region. These sequences, they are present in the constant region of heavy or light chain, right? So there are specific sequences that are present in the constant region of light chain or heavy chain, which will decide what class or what subclass. Let's say, for example, this is IgG1, subclass G1. There can be G4, A1 or A2. It will decide what class or subclass is that particular antibody belongs to. So these particular regions over here on the heavy chain and the light chain, which decides what class or subclass or what type or what subtype of light chain it is, are called the isotype. Okay, the antigenic determinants that characterize the class or subclass of heavy chain. That means here, for example, it is a class that is IgM. Here it is a subclass IgG1. And types and subtypes of light chain. Maybe it is here kappa, it could be lambda 1 or lambda 2. So these antigenic determinants which decides what heavy chain or light chain it is going to be are called isotype. And why? And that's what we talk about, right? 5 isotypes of antibody, that is IgG, IgM, IgA, IgD, Ig. These are the isotypes of antibody. And why is it isotype? Why is it same? Because if you take one particular species, okay, if you take humans, let's say, for example, all of us, all the members of this particular species would have all these 5 isotypes in the serum, isn't it? I would have, you would have, unless there is any immunological disorder or deficiency in a person. Otherwise, if a normal individual is there, in their serum, all of us would have these 5 isotype present in our serum. Since it is same for all of us in one species, it is same, it is called isotype. All right. So if you look at it, for human immunoglobulins, we have various light chains, various heavy chains. When they combine together, we get a particular type of antibody. And these regions, where are they present? Where is the location? It is on the constant region of heavy chain and constant region of light chain, right? These regions are present in the constant region of heavy and light chain. And where you will observe these antibodies, they are present in all the members of a species, okay? In all the immunologically normal members of species. So different species will have different constant region genes, right? This is for humans. For other species, the genes would be different. So they will have their own set of isotypes, which will be same again for that particular species. All right. So different species inherits different constant region genes and thus expresses different isotypes. Right. So if I take antibodies from one species and if I inject it in the another species, okay, if I take mice antibody and inject it into a human, then that human would induce the immune response against that particular antibody because it's from another species, it's a foreign molecule for that particular human. So there will be a production of anti-isotypic antibodies. Okay, those antibodies we will say are anti-isotypic. So that's an isotype. Now, what is the importance of this isotypes? How does it help us? The first thing that we know is based on this isotypes, what heavier light chain it is, we know what antibody it is, right? So it is very important clinically to understand what is the immunoglobulin levels of a person. In certain cases, if you want to know patients' immunoglobulin levels, what is the level of each immunoglobulin in the patient? So you can compare the levels of each antibodies based on the isotypes that are present, and you can predict, what could be wrong with the patient. And also you can look for the immunodeficiency, right? If you don't find any isotopes of IgA, that could be a very low level of IgA isotypes, that means there is immunodeficiency. So it's helpful for that. And also it is helpful in the detection of B cell tumor. Now this is very interesting. In the light chain, there are two types of light chain that we have, that is kappa and lambda. And there's no specific very strict ratio that what amount of kappa or lambda light chain could be there. But on an average, roughly, it is nearby 50-50 ratio. There should be nearby 50-50 kappa and lambda that is present in the serum of all the pool of antibodies that we have. But what happens in case of B cell tumor? It's a proliferation of a malignant B cell, defective B cell going crazy and producing so many clones of itself. So in case suppose this is a B cell which has the gene for kappa light chain, this has become tumorous, this has turned cancerous, and it starts multiplying. What you will have is the tremendously high amount of this B cell, the malignant B cell, which carries this kappa light chain gene. That means a lot of, you will observe tremendously high amount of kappa light chain in the patient's serum. So the ratio, which should be nearby 50-50, will be destroyed, and you will have a very high amount of kappa compared to that very low amount of lambda. Maybe it went to 9% and 10%. This means there is something wrong with some clones of B cell that is just proliferating. So there is the B cell tumor possibility. So this can help you detect the B set of genes that are going to express a set of heavy chain and light chain. Combinations of that gives us isotypes of antibody. But now when we talk about genes, we know that genes would have multiple alleles, right? Genes can have different alleles. That means there can be a variation in the genes. Genes that code for the same thing, but it will be slightly different. Let's say, for example, first let me give you an example. I'm talking about the hair color gene. I might have hair color gene, that codes for dark brown color hair. And you also would have that gene that codes for brown color hair, but your allele is coding for a lighter brown color hair. Okay, gene is the same, but there is a little difference in the sequence that is changing the amino acid in the final product. So what happens is, as I said, all of us will have genes that codes for these 5 isotypes. But it is possible that we all have different alleles for that, right? It is possible that what IgG I might produce will be different than yours because there is a little difference in the sequence. There is some difference in the sequence. All right. So that is called the allotype, and the Greek word allo means different. That is why it is called allotype. It is different. What is different? There is few amino acid change in the isotypes that we have. Let's say, for example, here the definition says antigenic determinants that are specified by allelic form of the immunoglobulin gene. Now we know that we have the 5 types of antibodies, but we might carry different alleles for that particular antibody. There might be few amino acid change in the molecule. Let's say, for example, this is IgG1 molecule from one person, and this is the regions, or these are the sequences that are expressed on the light chain as well as the heavy chain. In the same species, if I take another person and check for the same antibody, that is IgG1, there can be slight difference. As you can see on the light chain, there is slight difference over here. On the heavy chain also, there is little difference over here. Even though the antibody is same, the genes are same, but there is some amino acid difference. There is a little modification over there. That means the alleles are different. Such regions are called allotypes. Allo meaning different. All right, so these are allotypes. So allotypes are the determinants. All the members of a species would have same set of genes, same isotype, but will have multiple alleles. So they will have different amino acids. From one person to another person, allele will be different. So those regions are called allotypes. It can vary among the same species. So how can you produce allotypic antibodies? If I take antibody from one member of a species and inject it into another member of the same species, if I take antibody from you and inject it into my body, what will happen? My body will produce antibodies against this particular allotype, because it is something new for my body. And where are these located? Allotypes? Just as same as the isotype, because it's just the variation in the sequence. So constant region of heavy chain and light chain is where the allotypes are located. So it is observed when you do blood transfusion, even during pregnancy. If the maternal allotype is different than the fetus, because the fetus might have got the paternal allotype, which is different than the mother's allotype. The mother's body at the time of delivery, there'll be mixing of fetal and maternal blood. At that time, mother's immune system might produce some antibodies against the newly introduced allotype. But clinically, there is no problem even if we have some anti-allotypic antibodies. Anyways, so what is the importance of this allotype? How these allotype can help us? The first thing is very interesting. It helps us in monitoring the bone marrow graft. Okay. Now, when we go for grafting, there are a lot of things, tissue typing and a lot of other things that needs to be matched between the recipient and the donor. And that time, we can actually look for the donor allotype and can make a note of it. And when it is, the bone marrow is injected into the recipient, if the bone marrow is, if the grafting is accepted, the B cells that are present in the donor bone marrow will start producing the B cells, and there will be production of lots and lots of B cells that would produce plasma cells and giving rise to production of antibodies. Now, these antibodies that are formed from the donor's bone marrow would have that particular allotype, which is different than the recipient, right? If we have typed it before, we know what is the allotype of the donor. And if we find that in the bone marrow of recipient after a certain amount of time, that means the bone marrow graft has been accepted, and B cells have made, it has proliferated, produced plasma cells, produced antibodies successfully. So you can match the allotype with that of a donor. So it's an interesting way. It's not the only way, but one of the way to actually monitor the bone marrow graft. Also, paternity testing, which is not much of a use nowadays because we have DNA fingerprinting and all, sophisticated and very specific type of testing. But it could be helpful, or maybe it could be one of the tests to actually compare the allotypes. We know the allotypes of mother and father. We know the probable allotypes that a child might have, right? So we can compare that with the father and can check the paternity test. It's not that useful tool, or we cannot actually rely on this completely, but it used to be one of the helping tool for the paternity testing before we had all this DNA fingerprinting and all. And in forensics, it can help. These antibodies can be present in number of samples. It can be in saliva, it can be in blood. So you can take the sample and compare the allotypes with that of a suspect. So that way it helps in forensic also. So allotypes, as I said, allo means different. That means different alleles that are present in a given population. So again, we are talking about the two different individual of a same species. What happens is, let's move a little further and go to idiotype. Again, the meaning of word idio is own or very peculiar. Now, what happens when we encounter any antigen? The B cells, based on what type of antigen it is, will start producing specific antibodies against it. Very, very specific antibodies, because it will undergo affinity maturation. There will be somatic hypermutations, rearrangement of genes, resulting into very, very specific antibodies against that particular antigen. And this is because of the variable region, right? So in the variable region, there will be, that's why it is called variable region, which recognizes the antigen. Based on what antigen it is, it will have affinity maturation, there will be rearrangement of genes, somatic hypermutation, giving us it will result in the production of very, very specific antibodies against that particular antigen. Now imagine what antigens I might encounter during my life is not going to be exactly the same compared to anybody. My antibodies are going to be very, very specific, very, very peculiar for that particular antigen. So that is what is idiotype. Idiotypes arise from a sequence of heavy and light chains of a variable region, because that is the region that binds with the antigen, isn't it? It is the heavy and light chain variable region that binds with the antigen. So idiotype arises from the sequence of heavy and light chain variable region. When it encounters antigen, it is going to have somatic hypermutation, rearrangement of genes, and produce specific set of antibodies. So these are called the idiotypes. All right. Now, these idiotypes, let's say, for example, in my own body, I encounter antigen A and antigen B. They themselves would have, very specific idiotypes. Antigen A is different than antigen B. So I will have a set of antibodies very different for antigen A than compared to antigen B. So that is because we'll have different idiotype for that particular set of antibodies. So one B cell is encountering antigen A and another B cell is encountering antigen B. When they proliferate and produce a set of antibodies, they will be having totally different idiotypes than each other. This is going to be very specific and different idiotype than antibodies from this B cell. So idiotypes are the unique antigenic determinants that are present on the variable heavy and variable light chain region of individual antibody molecule. As I said, the antibodies from this clone of B cell would be totally different than the another clone of B cell, because they have encountered totally different antigen. So where will I find these idiotypes? As I said, what is the region where the antigen binds? It is the variable region of heavy and light chain. Sometimes it happens that it's not specifically only the antigen binding site that can act as idiotype. There can also be a site away from antigen binding site which could act as idiotype. Not necessarily it is typically only antigen binding site, because rearrangement of genes are happening in this variable region. So anyways, it can be any part of it. And where can we observe such kind of idiotypic antibodies? Where can I produce anti-idiotypic antibodies? If you take genetically identical twins, their isotype and allotype will be the same, right? Because they are genetically identical twins. But they will have different idiotype, because the idiotypes will be depending on what they are encountering, right? So that is not going to be exactly the same. No way it can be the same thing. So if I take antibody from genetically identical twin, from one twin to another twin, if I inject it, there will be no anti-isotypic or anti-allotypic antibodies because they are genetically identical, but the idiotypes will be different. So I will observe the anti-idiotypic antibody in case of genetically identical individuals. Now, how is this, idiotypes useful to us? How can these idiotypes help us? The first thing is it can help us in the treatment of B cell tumors. Now, how is that possible? Now we know any B cell, if it has become cancerous, it is going to have specific antibodies on its surface, right? All the B cells would have specific antibodies on its surface. And if there is a cancerous B cell that has just gone crazy and making lots and lots of its clones, all of them are going to have this particular abnormal idiotype present on it, right? There will be this specific idiotype expressed by all these tumored B cells because they are product of this particular clone, right? So if we can produce anti-idiotypic antibodies which are against this particular idiotype, very, very specific to this idiotype, we can target specifically only the tumor B cells, right? And how this anti-idiotypic antibody can kill these tumor cells? One is the natural calling of complements, and ADCC can take place. So one is that, and another very strong approach is labeling these anti-idiotypic antibody with some specific drugs or toxins that can bind with this idiotypes. Once it binds with the idiotype, it can actually lyse these tumor cells. Okay, that's how you can use the anti-idiotypic antibodies. Utilize the property of idiotype to kill the B cell tumors. It can be also used in the vaccine production. Now, this is very interesting. Let's say, for example, when there is an antigen, that antigen is going to produce a specific idiotype, right? That means antibody with specific idiotypes on its surface. Now, when your body encounters some antigen and it produces specific idiotype against it, think about it. As I said before, antibody itself is a protein. So now your body has made a new protein, a totally something which is new to your body. So that itself can also be recognized. This idiotype itself also can be recognized as an foreign molecule because it is something very new to your body. And it is possible that you produce anti-idiotypic antibody in your body itself, right? I hope it makes sense. When you encounter an antigen, which is absolutely a new antigen to your body, you're going to produce specific antibodies, specific idiotypes against that. And as I said initially, the whole concept of this antigenic determinants is because antibody itself is a protein that can act as immunogen. So these new idiotypes, they are very new to your body. That itself can be recognized as immunogen, and your body will again produce antibodies against these idiotypes. So that is anti-idiotypic antibodies. So there will be similarities between this anti-idiotypic antibody and antigen structure because they both can make complex with this idiotype, right? So if I want to produce a vaccine, I would not need an antigen, right? I can directly produce the anti-idiotypic antibody, which could act as surrogate antigen for me, which I can give it to a patient, and the patient would think this is an antigen and will produce specific idiotype against it. Now, why would I want to do this? In a scenario where the antigen is way too difficult, dangerous, toxic to be used as a vaccine. In that case, it is possible to produce anti-idiotypic antibody that could act as surrogate antigen. So that's how idiotypes can be useful in the vaccine production. So that's all. It's very easy to understand. Some key points that you need to know. Isotype, iso means same. That means all of us in one given species would have same genes for production of heavy and light chain. Since we are talking about genes, alleles also would come into picture. It is not possible that all of us would have the same allele. We might have different alleles for that given isotype gene. So those are called the allotypes, different alleles, the difference between your antibodies and my antibodies. Here's the allotype. And if you go furthermore in detail within your own body, you will have very, very specific antibodies based on what a B cell is encountering. So furthermore, you will have your own set of every B cell which encounters a different molecule would have very specific idiotypes. And because it is the part, variable region is the part where the antigen interaction occurs, these lies in the variable region. So that's all. That's